Okay, good, good. Okay, good afternoon, everyone. Thank you, Jushin, for the kind introduction. My intent is not to convince you that sepsis is a metabolic disease, but to introduce this idea that there's a component of sepsis that is metabolic and that we can exploit that metabolism to identify prognostic and predictive biomarkers and potentially drug target opportunities. All right, let's see if I can get this to work.
Use the mouse to click first.
Okay.
Use the mouse to click first, then.
Or just maybe. Okay. I have no financial conflicts to disclose. So this afternoon, I'm going to start by just giving you a brief overview of my research program for those of you who might not know me, discuss sepsis and its impact on human health, which many of you might not appreciate how significant that is, review the science of metabolomics and pharmacometabolomics, and then how we have utilized those sciences to phenotype sepsis and therapeutic response, and then present some options or ideas for candidate therapeutics, some of which already exist. They're known drugs that might have therapeutic value in sepsis. So I run a research program in translational metabolomics and critical care, and there's a lot on this slide. So I'll point out a few things. Despite it being in critical care, our focus is primarily sepsis and ARDS, which affects over 1.5 million people in the US each year. Despite that impact on human health, these diseases are vaguely defined, and there's really no targeted pharmacotherapy. Most of the therapy for these diseases are supportive. And proteomics and genomics, which have been utilized for decades, do not fully explain the heterogeneity of these diseases, nor have they been successful in identifying paradigm-changing therapeutics. So we use a multi-pronged approach. We employ metabolomics, measuring small molecules and biological samples, and I'll talk a little bit more about that. But we also use both porcine and murine models. We have a swine model of sepsis and ARDS, which we are testing different therapeutic strategies in. We also still use murine models of sepsis. We think there's some value, and I'll show you some data we've generated from a mouse model of sepsis. And we utilize biospecimens, mostly blood specimens from patients with sepsis and ARDS to translate our work. And our long-term goal is to identify and test biomarker-directed pharmacotherapy to advance therapy in these critically ill diseases or critical illnesses that right now have very few effective pharmacotherapy options. And our work has been funded by the National Institutes of General Medical Sciences at the NIH. So for those of you who may not be familiar with sepsis, it poses a significant threat to human health. And these are just some statistics that I'm going to put in context for you with a disease that you might be a little more familiar with because it's gotten a little more attention. But every year, at least 1.7 million adults in the US get sepsis. So our work is focused on adult sepsis. This does not even include pediatric sepsis, which is kind of the other extreme of the sepsis bell-shaped curve, if you will, kind of the two extremes of human age, very young people and older people compose this. But that's not to say that young, healthy people don't get sepsis. They do. Of these patients, 270,000 of them die each year. And remarkably, 1 in every 3 patients who die in hospital die from sepsis. So this has an extraordinary impact on human health. You may be a little bit more familiar with myocardial infarction or heart attack because over the years, it's gotten a lot more attention because it has a hallmark symptom. The hallmark symptom of MI is crushing substernal chest pain. So that is a significant advantage for myocardial infarction. And I'll talk a little bit more about that as I go in the talk. But just in context of what sepsis means in relative to a disease that we know it is a significant hazard to human health, MI. Sepsis we have 2.5 times more cases of sepsis than we do MI in the US each year. The hospital mortality rate of septic shock, which is the most severe form of sepsis, is 8 times higher than the most severe form of MI. So it kills a lot of people. And from a healthcare cost perspective, sepsis is the most expensive condition treated in the US, approaching close to $24 billion, that's a B, billion dollars a year, which outpaces, with all due respect to our oncology colleagues, cancer. So this is really a large health problem in the United States. And these trends are similar across the world. So despite these striking statistics, sepsis remains a quandary for precision health because the diagnosis is very inaccurate and incomplete. The diagnosis is often vague because patients present with vague symptoms. There's no hallmark or pivotal event that occurs. Patients will say, well, I was home, I wasn't feeling well, and now I have this fever that just won't go away. I'm not feeling good, so I thought I'd come to the emergency room. So there's just no hard and fast diagnostic symptom that identifies sepsis. There's also just a paucity of prognostic and predictive biomarkers that help us make an accurate diagnosis. For example, in MI, the electrocardiogram, we can measure the electrocardiogram and be pretty confident about a diagnosis. And then we partner that with blood levels of specific biomarkers that we track over time that don't just help us diagnose, but help us prognosticate. And these the culmination of these shortcomings make treatment for sepsis very imprecise. So we are aggressive with antibiotics, which is kind of our first line of intervention. But after that, care is primarily supportive. We support blood pressure because these patients can rapidly progress to shock where their blood pressure drops, and we need to employ what we call fluid resuscitation, or in some cases, vasopressor support to maintain blood pressure. And typically, outcomes can be not just poor in hospital, but in patients who survive sepsis and get out of the ICU and out of hospital, many of these patients can have prolonged and persistent problems that range from cognitive dysfunction to mobility dysfunction. And I think COVID really highlighted that, what we see with long COVID is really not unlike what we see with a lot of patients with sepsis. So with all that vagary, the definition of sepsis follows suit and is also vague, and it certainly does not reflect the complexity or the heterogeneity of the illness. The third international conference or consensus definition is one of the most commonly used definitions of sepsis, and it defines sepsis as a life-threatening organ dysfunction caused by a dysregulated host response to infection. Vague. An organ dysfunction in of itself is often identified by a score. We like scores in critical care. We like to add things up and come up with a score. In this case, we use the sequential organ failure assessment score, which takes into account function of the major organs. And we assign those measurements points. And the higher your higher your SOFA score, the more severely ill you are, and the more impaired your organ function is. So as sepsis progresses, increases in that SOFA score are indicative of worsening disease. And then septic shock, which is the most severe form of sepsis, has a few more additions to it, most notably the use of lactate, a metabolite, in its definition, and patients who have a serum lactate of greater than 2, despite adequate volume resuscitation, are classified as septic shock. And these patients are most recognized by their hemodynamic instability. So their blood pressure is unstable, they require not only fluids, but often vasopressor support to maintain their blood pressure. So just to put it very simply, kind of in a layperson's term, sepsis occurs when your body develops an overwhelming inflammatory response to an infection. And this is really the general definition, and it doesn't go much further than that. We have two primary tools that we use clinically. One is the blood lactate level. Typically, the higher it is, the more severe or worse the sepsis is. And the sequential organ failure assessment score, the higher that is, the worse, the more severe the disease is. So with that in mind, and particularly given the inflammatory nature of sepsis, you would think that anti-inflammatory agents would be effective for the treatment of sepsis, and that has not been the case. There's a graveyard of failed sepsis therapeutics. These are just some examples that have failed in clinical trials. And what's I think very disconcerting about this is that this continued failure of randomized clinical trials in sepsis has really jaded pharma from pursuing drugs that might have utility in sepsis. So it's really left us without any pharmacotherapy armamentarium to treat sepsis. So more recently, we've recognized the importance of the heterogeneity of the disease. And there's a lot of different components of sepsis that seem to result in varied responses. So for example, the immune response can be highly varied. The hemodynamic response can be highly varied. Temperature trajectories can be highly varied, and end organ function can be highly varied. The extent of metabolic derangement can be highly varied. And even in adults, we know that the incidence of sepsis increases with age. Age is an independent variable or factor for sepsis survival, but it's not the only predictor for sepsis survival. The relationship with sex is not as clear. There's really mixed evidence whether males or females have differences in mortality. And then again, to make matters even worse, there's really no true time zero. And this is important because like myocardial infarction, we know that the sooner we treat patients, particularly with antibiotics, the better they do. So not having that hallmark time zero sign or symptom has really hindered our ability to manage these patients. So with that in mind, a number of different groups around the world have been trying to leverage this heterogeneity, all these different variables that move at different rates in these patients to subphenotype and identify subgroups, if you will, of sepsis that might help us pinpoint potential therapeutic targets. And there's a number of different approaches that have been used. I'm just going to show you some examples. One is a study that was done using blood transcriptomics of immune gene expression. And this group found two different signatures, one that was a neutrophilic suppressive signature and another one that was an inflammatory signature. Interestingly, they used healthy controls as their comparator group. Another larger study used a retrospective analysis of electronic health record data and identified 4 clinical phenotypes. They also looked at serum biomarkers of host response when they developed their models, and they found these four distinct phenotypes that were related to organ function: hepatic, renal, pulmonary, and cardiovascular. So kind of aligning with the SOFA score measurements. But this model has been called a hot mess. And really, these kind of approaches have been called hot messes because if you think about the clinical situation, it's a medical emergency. Sepsis is an urgent situation. And to do all this information gathering takes time. And the longer it takes, the longer you're prolonging or delaying potential life-saving intervention. So while they're informative of some mechanisms, they're not really clinically useful or implementable, if you will. So we've taken a different approach. We've used metabolomics to understand sepsis heterogeneity. And one of the first steps that we've taken is to look at this approach for biomarker discovery. And for those of you who may not be familiar with metabolomics, it's a member of systems biology, along with its colleagues, the genome, transcriptome, and proteome. And the advantage of the metabolome is it's like that upper level read of all the activity of its colleagues. So it does give a broad read. The challenge of metabolomics is because it has this broad read, it's very dynamic and it can fluctuate rapidly over time. Even if you don't know about metabolomics specifically, if you're a clinician or you work in the clinic, you probably know more about metabolomics than you might appreciate, because 95% of all our current diagnostics are small molecules. We measure small molecules in the clinic. This includes serum creatinine, cholesterol that we use routinely. We also know that 30% of identified genetic disorders involve diseases of small molecule metabolism. And small molecules serve as signaling molecules and cofactors for a number of different protein pathways. Interestingly, despite the attention on biologics recently, 90% of all the FDA-approved drugs and 75% of the approved drugs in 2020 were small molecules. So the other advantage of metabolism and looking at small molecules is that metabolism is actionable. And in fact, in critical care, we often say we have oncology envy, because oncology has exploited metabolism for decades. Many of the chemotherapeutic agents target metabolism. So just as a base definition for metabolomics, metabolomics is defined as a simultaneous measurement of small molecules, most often defined as a molecule less than or equal to about 1,500 Daltons in a single biological sample. So unlike the single measurement that we might make for serum creatinine, when we assay a blood sample, we assay it for multiple metabolites at the same time. However, we cannot capture all of the metabolome by a single analytical platform. Two of the most common analytical platforms are LC-MS, which gets a pretty broad read depending on the types of columns that are used, and NMR, nuclear magnetic resonance spectroscopy, which is not as sensitive as LC-MS, tends to be more quantitative. LC-MS metabolomics is often referred to as untargeted metabolomics, so it gets a broad relative intensity read of a lot of different classes of different compounds. NMR is like the 35,000-foot view, measures more high abundant compounds. And there's other analytical approaches as well. But the thing to appreciate is that no one analytical platform captures everything. And even by using combined platforms, we probably don't get everything. And I would also say that metabolomics really refers to small polar molecules rather than lipids. Lipids is a domain in and of itself called lipidomics, which I will not talk about today. I think Dave Wissert, who is at the University of Alberta and one of the renowned investigators in metabolomics, another way to think about metabolomics in the context of systems biology is that genomics is what may happen and metabolomics is what just happened. So when we think about sepsis, we think about it in a little bit different way than maybe it's been traditionally viewed. And to think about it from the metabolic underpinnings of sepsis and what's actually happening in the host, and some of the energy economies that exist and compete with each other during sepsis. So the first is the culprit pathogen. So the host is being inundated with an infectious pathogen, and this culprit pathogen is competing with the immune system and the host for nutrients in order to multiply and grow. And so it is utilizing nutrients itself. The immune system and the immune response, invading pathogens can use metabolism to outsmart host immune response. And then we know that rapid escalation of the immune response is important for an efficient response to the pathogen. But we also know if that's too exaggerated, that can be detrimental. So either way that immune system is using energy to manifest that response. The other is the gut microbiome. The gut microbiome competes for host nutrients, and it both produces nutrients and uses nutrients from the systemic circulation. And where we focus most of our effort is on host metabolism, particularly at the organ level. The energy economy of the host is severely disrupted by sepsis, and it may not completely recover. And I'll talk about that a little bit later in the talk. And we also start to think about each organ and the economic energy needs for each organ could be different in sepsis. And then, you have a competition between nutrient and energy substrates across organs. We often use the blood as our primary biofluid. So we need to appreciate in measuring this biospecimen that takes a physiological average of all these systems, it does reflect all these different systems. And so sorting this out can be quite challenging. I've intentionally put lactate here because I want to highlight that they're not unique substrates that these different economies are using. They're all competing for similar fuel substrates, including lactate and glucose. Some of them may produce different things, like the gut microbiome produces short chain fatty acids, but the competition is on, and this creates an energy crisis for the host. We have leveraged a clinical trial that was conducted by Alan Jones at the University of Mississippi to better understand the metabolic consequences of sepsis. And this was a randomized clinical trial, a phase 2 trial of L-carnitine versus placebo in patients with vasopressor-dependent septic shock. So they were very sick. And we utilized the blood samples that they collected as part of the study. So this study was a randomized trial, as I mentioned, and then patients were randomized to placebo, or they received one of 3 different doses of L-carnitine in a adaptive dose finding design, and blood samples were collected before treatment and then 24 and 48 hours after. We assayed the serum for using NMR, and we assayed them for acylcarnitines. And we used these data to identify differentiating metabolites of mortality initially based on the time zero values. And what we found, at least from the time zero measurements, the most differentiating metabolite was acetylcarnitine. And this is the differentiation in 28-day survivors and non-survivors. This was sustained in patients who were, I don't know, Dushan, can you close that, or can I close that just because it's blocking the slide, sorry. There we go. The placebo-treated patients, as well as the L-carnitine treated patients. So this was time zero, but this was evident across both of these patient groups. We also found that, oh, this didn't translate very well. There was a nice color there, but that lactate, which we know correlates with SOFA score, correlated well, which is what we expected, but we also found that acetylcarnitine correlated very well with SOFA score. But we also found that patients elevated lactate levels that were mitigated even in non-survivors. Those patients with elevated acetylcarnitine levels, so these were 28-day non-survivors, was not mitigated. So what we do in sepsis, we're mitigating these elevations in lactate, but we're missing a metabolic component of sepsis because the acetylcarnitine levels in non-survivors remain elevated over the course of sepsis. We also looked at chronic critical illness. So these are patients who survive sepsis, are discharged from the ICU. And this was a secondary analysis of race study participants who received placebo required mechanical ventilation and had a blood sample within 36 hours of the initiation of mechanical ventilation. And we looked at their time to extubation and removal from vasopressor support. And what we found is that the two most differentiating metabolites were acetylcarnitine, measured by both NMR and LCMS. To think of this in a little bit different way, we dichotomized the acetylcarnitine measurement. Patients who were at the median or above were categorized as high acetylcarnitine, and those below it were categorized as low. So this plot shows the proportion of patients that had high acetylcarnitine, which is the red line. A lower proportion of patients who had high acetylcarnitine were survived to be extubated and shock-free during their hospital course versus patients with the lower acetylcarnitine, suggesting that acetylcarnitine, and particularly the persistent elevation of acetylcarnitine, may be a biomarker for prolonged, what we call chronic critical illness, or just a delayed recovery in patients with sepsis. So I know it's late in the day for biochemistry, and full disclosure, I am not a biochemist, but just to highlight how acetylcarnitine is being produced. First and foremost, it starts with L-carnitine. So L-carnitine is an endogenous metabolite. We derive it primarily from our diet, but it's highly conserved. It's required to shuttle long-chain fatty acids into the mitochondria, which through a series of enzymatic steps end up with the production of acetyl-CoA. Acetyl-CoA is required for the TCA or Krebs cycle, if you learned it that way, and ultimate generation of NADH for the electron transport chain. We think that in sepsis, because of the high energy demand, there may be a TCA cycle stall. We know that there's some disruption in beta oxidation that ultimately could lead to excess acetyl-CoA that the mitochondria needs to get rid of or dispose of. So it's possible that in the presence of L-carnitine, that this drives the formation of acetylcarnitine through this carnitine acetyltransferase pathway. Acetylcarnitine is then shuttled back out into the systemic circulation. It's in equilibrium with L-carnitine. It gets converted back to L-carnitine, and it starts all over again. This is an important metabolic mechanism for long-chain fatty acid metabolism. And just to highlight that one of the key features of acetylcarnitine as a biomarker is that there's no subsequent downstream signaling. So unlike lactate, which I'll talk about again in a few minutes, there's no other downstream signaling for acetylcarnitine. It's really contained within this pathway. We are not the only ones to make this finding. There's been a number of studies that have identified acetylcarnitine as a predictive marker of sepsis severity. So we are working with many of these groups to better understand this process. And we also know that lactate, even though we think it's an important part of our understanding of sepsis and what is metabolically going on with sepsis, it does not fully reflect the energy crisis of sepsis. It is still the most common clinical biomarker for sepsis prognostication, was just to highlight some of its limitations. Even modest elevations in sepsis, kind of in the 2.5 to the below 4 range, have been attributed to mortality, even in patients that don't require initial hemodynamic support. So these would be modestly ill patients and patients without any sort of end organ dysfunction. It used to be viewed as kind of the hypoxic waste product. I think that was the really initial idea behind lactate, and that it was indicative of hypoperfusion to end organs. But we know that that's probably not the case, particularly since we know that we can mitigate those elevations in lactate and really have not only modest improvement in sepsis mortality, but modest improvement in organ function. I think recently, and I could spend an hour just talking about lactate because I think lactate's fascinating. I think more recently we've come to appreciate the promiscuity of lactate. It does a lot of things. It interacts closely with the mitochondria. There's controversy here, but it's likely doing many more things as a metabolic mediator and as a fuel source than we currently appreciate. So in addition to our work in biomarker discovery, we've also leveraged pharmacometabolomics to understand the therapeutic response to sepsis. And I would just like to make a pitch about pharmacometabolomics. It doesn't seem to be that well known, but I will argue that it is an important member of kind of the pharmacology enterprise. We've viewed it as not just a predictive approach, but also a way to assess drug response. And in that, we think it's an important partner with other systems pharmacology like pharmacogenomics, PK, and PD, so that it could really be informative of mechanism of drug action, but also differentiating different types of response across patients. So again, for this work, we leveraged the RACE trial. We used the same data. We used our serum metabolomics data from our NMR and acetylcarnitine assay. And in this case, we focused our efforts on the 18-gram treatment group. So from the RACE outcome, it did not meet the predefined goal, if you will, of superiority for SOFA score reduction, but there was a modest reduction in mortality with the 18-gram dose. So this did catch our attention and curiosity, and Teddy Gennaro and Chris Gillies of the Weil Institute. So Teddy is a graduate student. He defended earlier, and Chris Gillies, a data scientist at the Weil. He's now at Regeneron. Spearheaded this effort to really understand how or if there is a specific metabolic phenotype that might be responsive to L-carnitine treatment. So in this study, we looked at the cumulative fraction of deaths within the RACE trial, and we typically use 28 days in mortality in sepsis, and it's somewhat arbitrary. So we actually looked at the cumulative fraction of deaths in the study, and 90 days is where we actually hit the plateau. So we used 90-day mortality in this analysis. We constructed a series of regression models, and we found that when accounting for age and SOFA score, there were 9 metabolites that discriminated 90-day mortality, and acetylcarnitine lit up again, and this analysis withstood Bonferroni correction for false discovery. We then accounted for dose, the interaction model, which is accounted for dose and against metabolite, and again adjusted for age and SOFA score. This model did not tolerate adjustment for multiple comparisons, but the metabolites that did have significant p-values included several acylcarnitines, one of which was acetylcarnitine. And probably one of our most profound findings was in both of these analyses, lactate did not light up. Lactate was not significant in either of them. We then followed this by constructing an analysis to identify of the most significant metabolites, which threshold concentration, was there a specific concentration of metabolite that might be driving the separation and the benefit from L-carnitine. So again, we focused on the 18-gram dose of the survivors and non-survivors, and constructed a series of Z-score analyses and set cut points based on, for this case, acetylcarnitine concentration. So this is the whole cohort showing that there's no difference between placebo and the 18-gram dose of carnitine. But as we increase the concentration of acetylcarnitine, we could improve the mortality benefit. So this was just a proof of concept analysis to suggest that there's metabolic differences within the acylcarnitines, in this case, acetylcarnitine, that could differentiate the mortality response to L-carnitine supplementation. So to think about this in a different way, because I think one of the drivers of failures of clinical trials in sepsis is that we're taking a very heterogeneous mix of patients and we're just studying them all together and we give them all the same drug, or we randomize them to placebo or the drug, and don't take any other factors into consideration. If we could use a pharmacometabolome approach to develop an enrichment strategy for patient enrollment into a clinical trial, it could look something like this for L-carnitine, where you have patients with septic shock, you measure their acetylcarnitine concentration. For patients who have an acetylcarnitine concentration that's greater than or equal to 30 micromolar, those patients get randomized. And so that would enrich the trial with potential responders to the drug. And we think about what we need to do differently for this group of patients. So just for exercise, we went back and said, oh, well, what happens if we'd done the race trial this way? What would it have looked like? So this is the acetylcarnitine concentration. This is patients who were randomized to either carnitine or placebo. At time 0, they had a acetylcarnitine concentration of at least 30 micromolar. And for the carnitine treated patients, their 28-day mortality would have been 58%, versus the placebo patients would have had a mortality of 88%. And admittedly, these are small numbers, but it introduces the concept that we could use a pharmacometabolomic strategy to identify patients who might be more likely to respond to a specific therapeutic, particularly a metabolic intervention like L-carnitine. So we're very curious about what is driving the acetylcarnitine differences in the broad range of acetylcarnitine concentration in patients with sepsis. So we go back to a little bit of biochemistry and the carnitine acetyltransferase enzyme. And to generate acetylcarnitine, we need carnitine and acetyl-CoA. And we have hypothesized that part of what's driving this is there's this excess of acetyl-CoA that the mitochondria just can't use. And so they need to dispose of it. And by providing a surplus of carnitine, we can drive this reaction. So we are trying to use some experimental models to try to untangle this mechanism a little bit. And these are very preliminary data. These experiments were done last summer by an undergraduate student in our BSBS program, along with Mark McCann, who's here this afternoon. And we induced a polymicrobial sepsis in mice. And we did this by a cephal ligation puncture. So this induces a systemic sepsis that systemic infection that impacts globally the organs in the animal. We gave an early intervention in some of the mice. 6 hours after that, they either got a large dose of carnitine or saline placebo. And then we also did this late intervention. And we weren't interested in mortality. We really wanted to look at what was happening at the organ level. And so we ended the experiment at 30 hours. And we did a procedure called a freeze clamp of the organ, and then we did an NMR measurement of metabolites in that organ, and then we measured tail vein whole blood by LCMS for acetylcarnitine. And this is kind of what it looks like. We use this freeze clamp procedure, and so I use the heart because the heart, I can animate it, it moves. And the key to this is that we want to rapidly quench metabolism. So we do that using what we call this freeze clamp procedure. So we use these tongs that have been in liquid nitrogen, so they're freezing cold, and we bio-squeeze them in these special little baggies, and we make sure everything is really, really frozen. So we pancake the organ essentially, and that's an advantage of the mouse because it's got a relatively small heart, and we can do that. We collect a blood sample, And then we process that organ sample and we assay it by NMR, and we get about 45 metabolites. But the metabolites we get are of great interest to us. So we get a good read of the adenosine pool, we get acetyl-CoA. It's not on here due to space, but we get carnitine, acetylcarnitine. We get a nice read of those mitochondrial metabolites and some of the TCA metabolites. And so what we found is that when we looked at the animals that got L-carnitine early, we calculated the carnitine acetyltransferase enzyme score just by looking at the products versus the substrates, that ratio. And we found, not surprisingly, that in the L-carnitine animals, some of my text isn't lighting up on the screen for some reason, but this was driven, probably not unexpectedly, by increased carnitine because of the supplementation. And then later, it shifted. And this was driven primarily by an increase in acetyl-CoA and acetylcarnitine. What was interesting, though, when we looked at energy balance, in the early measurement, there was no difference. But in the later measurement, there was this shift towards improved energy balance in the L-carnitine treated mice. So at least in the heart, and we bias this a little bit because we use the heart because it's loaded with mitochondria. So we worked in our favor maybe a little bit, but suggesting that carnitine supplementation may be advantageous in the setting of sepsis. We still had to do quite a few more experiments, as you might expect, just to get our N values up and also to test different time points. But these data do suggest that in the presence of L-carnitine supplementation in this excess L-carnitine environment, that the acetyl-CoA level may be important in driving the production of acetylcarnitine. And it supports the idea that that magnitude of elevation of the acetyl pool may be important for eliciting a therapeutic response to L-carnitine. And this may explain why patients who have this high acetylcarnitine level may be responsive. So it seems that there's some suggestion that L-carnitine supplementation could improve bioenergetics, at least at the cardiac level. And we're starting to think about sepsis more from just specific organs and thinking about, there's some patients that may have a similar SOFA score, but in one patient, that SOFA score could be driven by the cardiac SOFA score, versus in another patient, it's driven by the renal score or the liver score. So there's that level of heterogeneity across SOFA score measurements because we typically bundle those together. So we also have the other organs that we have in our freezer that we're getting ready to assay that hopefully will also provide more insight about this. So I think that collectively, we're interested in how these metabolic signals could solve some problems of sepsis heterogeneity, advance our understanding of what drives organ dysfunction. So we could think about how we might preserve post-organ function, particularly in terms of bioenergetics, and then maybe support the mitochondria through the energy crisis of sepsis that could be done possibly through drugs, and thinking about the mitochondria as a druggable target. And there are some mitochondrial and metabolic modifying interventions that are already out there. Some that have been tested and not done well in other diseases, but may be repurposed for some of these opportunities that might exist in sepsis. So we're thinking along those lines. And there's also, and some of these have been tested in animal models. The Zephyr has, and there is a paper that they did mitochondria transplant in the lung for sepsis-induced ARDS in a mouse and showed improved lung function. So there's some hints that this might be not only feasible, but could be a new avenue of therapeutics for sepsis. I also think there's additional considerations that we need to think about for the ICU. So I'm a critical care person. So I've worked in the ICU, and I kind of joke with my colleagues now that I've been out of the ICU for 15 years, but I could go back in and it wouldn't be any different. The same as it was, because we're doing the same thing as we've been doing for decades and decades. We measure the same analytes in the ICU that we do in healthy people, which makes no sense to me whatsoever. I kind of joke about drug dosing in the ICU. We used to stand at the end of the bed, and if the patient's head fit in your thumb, it was like 0.25 of digoxin. If the head came around your thumb, it was 0.5. Like, no pharmacokinetics here, right? Because these patients are so volatile, their organ functions are changing rapidly. We don't measure those organ functions at the granular level. We tend to be reactive instead of proactive. And this is really the quandary of critical illness that hasn't changed for decades. So things like nutrition, I can tell you that nutrition in the ICU is all over the map, depending on where you land. There are some advocates of it. Others say, no, that, it supports infection. You shouldn't feed patients. So I think understanding what the metabolic needs are of our patients in the ICU could maybe help us tailor these regimens to a more specific target. We also don't measure some really important nutrients and cofactors that could maybe help more granularly phenotype these patients, particularly in the elderly, where we know that some of the B vitamins have dropped off just with age, and they may not have those cofactors. So they may have the fuel substrate, but if they don't have the cofactors to drive the enzyme, it's not going. So there's things like that that we don't even think about or do routinely in the ICU. There's a lot of interest in early mobilization because we know that patients who are in bed long, particularly in ICU where they get a lot of sedation and paralyzing drugs, they atrophy their skeletal muscle. And that has metabolic consequences. It also has functional consequences. There's actually some data that suggests that too early mobilization is actually detrimental in these patients. So understanding that and the biology of skeletal muscle reserve and skeletal muscle function and how we might improve that and preserve it. And I think then there's the whole other piece. I work with a colleague in the microbiomes. That's a whole nother realm that certainly interacts here. We're still learning about antibiotic use. Crazy stuff with antibiotics, and what we are carpet bombing the gut microbiome with our aggressive use of antibiotics, and that in of itself could be problematic for particularly critically ill patients. And I don't want to dismiss proteomics and the immune system itself, which still is very important in sepsis. So thinking about the metabolic phenomenon related to sepsis as a component of sepsis, along with the immune response and the hemodynamic response. These are a collective set of processes that are occurring simultaneously and in parallel. So with that, I have to come to my important acknowledgements. I have a small but mighty lab that has had the privilege of two outstanding graduate students over the last 5 years, Teddy Gennaro and Mark McCann. Both have successfully defended their theses. And Thomas Flout, who is the master of freeze clamping, Laura McGowan, who manages the lab and does a ton of work as well. Bo Dong is the undergraduate student that spent last summer in the lab and did the freeze clamp of the heart, and Larissa. Well, so I am not an NMR spectroscopist either, and Larissa keeps me honest. So she's like, no, Kathleen, no. So that's really important. I've learned tremendous amount from all of these extraordinary individuals. And if you want to find out more about my research program, it's there. I work very closely with the regional core. we have an outstanding metabolomics core. We do a lot of the NMR components, and they do the LCMS component. We collaborate on a lot of things, do a lot of validation work. So it's wonderful to have those collaborations. My colleagues in crime in PCCM, Ted Staniford, who's the former chief of Pulmonary, who's since retired, really took me under his wing and taught me a lot about critical care research. And Bob Dixon, who's extraordinary and is a microbiome guy. And so I've learned a lot from him, and along with Mike Schoding and Liz Vigilante, who Liz has worked, she's really interested in chronic critical illness. So she worked with us on the chronic critical illness story for acetylcarnitine. And then my colleagues at the Weil. So the Weil is spearheading large animal work in critical illness. So we have these pig models, and that's been a lot of fun. And I'm going on sabbatical in July to dive into that. So I'm excited about that. And then I would be remiss if I didn't point out my Alan Jones. I met Alan Jones over a FaceTime call almost 15 years ago, and we collaborate to this day. And Alan sent me his blood samples from his clinical trial. He didn't know me from Adam. And the rest is history. So just like a serendipitous sort of interaction through another colleague. And Mike Bouchard, who actually was here last month. Mike is also a longtime collaborator. He was a former fellow of Alan's. He's now at the University of Minnesota, still doing great things. And grateful for the NIH for our funding. So thank you very much for your attention. I'd be happy to answer any questions.
The online attendee write your question on the either chat or Q&A session. We can have a question from the audience here. Tim.
Thank you, Kathleen, for that really fascinating talk. I found myself wondering at the phenotypic level, if you did a post-mortem on the patients, does it look homogeneous in that way, that you find like a big, I don't know, occlusion of bacteria inside, you just can't see it with the tools?
Yeah.
Or is it very heterogeneous?
Well, that's a great question, Tim. It's very heterogeneous. So if you think about just infection, so we use sepsis kind of as a metric of infection, but it's not source specific, right? So there's patients who have infections in their kidney, pyelonephritis, and the bacteria is in the kidney, but it's just seeding the blood. One of the most common causes of sepsis is pneumonia, and there's a whole bunch of different types of pneumonia, everything from viral pneumonia, COVID, gram-negative, gram-positive pneumonias, abscesses. It's just anything that can cause an infection can lead to sepsis.
So maybe as a follow-up to that, would we think about a target for sepsis if if such a thing could exist in the same way that we think about targets of cancer, that we'll need 100 different drugs for sepsis in the future?
I think we might need 100 different drugs for sepsis. Yeah, that would be like my dream. Like that we could be in the ICU and say, oh, we're going to use this on this patient, we're going to use that on that patient, right? Because I think what cancer has done, what oncology has done, is they have granularly phenotyped cancer to target specific therapies. Could we get that granular with sepsis? It might take us a little bit, but I think we could get more granular than where we are right now. Like, to me, we're like at 35,000 feet. We need to get more granular and really understand the mechanisms that are driving this, but also make it clinically feasible to rapidly phenotype these patients, right? So I don't want to, I called it a hot mess, and actually I stole that from a colleague of mine. But like, those are still informative. We're still learning something about these signals, but to implement those phenotyping schemes in the clinic is labor intensive, cumbersome, and not really clinically practical. We can't implement them.
Question from Deb online.
Oh, Oh, there's questions online.
Last one.
Okay. We can hear you. Chat disabled. So you have looked at, have you looked at dihydrofolate reductase path in relationship to L-carnitine and or influence in methionine as well? Deb, that's a great question. We have not. So one of the challenges with metabolomics is, one, we're measuring stuff in the blood. So we don't really know what's happening at the cellular level. And a lot of those metabolites are very low abundant. So it's hard to pick them up. We do get methionine, but getting, enzyme activity is a kind of a different animal to tackle. But it's a great point. There's still a lot for us to do, certainly.
I have a question. Acetylcarnitine perhaps is a marker. That's a
Yes.
damage, right? Sepsis is so messed up in the body. Perhaps there's no single target to say, hey, there's a drug target, a single thing you can have. Perhaps
Right.
everything going on at the same time.
Right.
Has anybody thought about like use the acetylcarnitine as a readout, just to screen some kind of mixture of things, nutrition in the cell line,
Right.
to figure out some of the combination, either existing drug or existing nutrient to see, hey, if I can reduce the acetylcarnitine, maybe that's the good cocktail of a therapy.
Yeah.
Anybody ever done that?
I don't know if anyone's done that, Dushan. That's a great idea. A cell model of sepsis. We've picked that around like a cell model of sepsis to do like rapid screening of metabolic reads and then, throwing drugs in there and seeing what happens sort of thing. So I think that's something that we need to consider. One of our challenges is that NIGMS is very focused on studying human sepsis, which is challenging. We think that there's room for using human sepsis and then a little bit iterative process to go back to the lab to look at, still utilize urine models in some cases to answer specific questions. We think that our pig model will enable us to get more, serial blood samples over time from a single animal, get tissue and things like that. But I think it's going to take a multi-pronged approach, right, to tackle this and to change the clinical paradigm to go beyond just measuring lactate and glucose. Is what, like, those are our favorite biomarkers. Like, well, we've been doing that for a long time. And where has it gotten us? Hasn't gotten us very far. So just kind of rethinking these components that might be informing us of other processes that we're missing, that we could target. And I think, lactate is so interesting. And I think that whole story has just changed dramatically, that we're all very interested to see what the consensus definition will be about lactate, because it may not be the informative thing that it was, or it's just telling us something different.
I mean, that was.
So, my question, I have a couple of questions, but I'm going to try and keep them brief. But the first is just this idea of like sepsis and what the way we look at it in the clinical setting. It starts off with this idea of recognition of fever, right? So it's sort of this ramped up metabolism in a sense, right? How sensitive is metabolomics to body temperature?
It's a great question. I think there are data out there just looking at body temperature and metabolic rate. Like it's a metric of metabolic rate, but the fever response to infection is highly variable. And my colleagues in PCCM have looked at this and shown that, temperature trajectories are all over the map. And some of the sickest patients who have the poorest outcomes are patients that don't manifest fever. Do I know that they're not as metabolically perturbed or less metabolically perturbed as maybe people have higher fevers and have a better outcome? I don't know that. We haven't gotten that far yet. So we're hopeful to get a grant, a notice of award, hopefully soon, for a large consortium grant to study patients with sepsis. It's going to be an ARDS pneumonia sepsis cohort across 5 centers. And we're going to study 5,000 patients longitudinally. And we're going to follow them out to at least a year. Hope more. I've roping Tony into this. So that we can learn more, that we can answer some of those questions in sepsis.
Thinking about your study with the base trial and this ideas of dosing L-carnitine, part of the challenge with L-carnitine, as you pointed out, is it depends on the pool. The largest pool is a skeletal muscle. And so perhaps some patients that got 18 grams maybe needed it more, let's say, than other patients that might have had more muscle mass and needed less. But kind of associated with that, because it's related to skeletal mass and diet potentially. I think what I've read is like vegetarians have like maybe 10% less L-carnitine than omnivores
And vegans.
and vegans, and
Yeah.
compared to, carnivores. Obviously, really difficult to do this kind of study design. We're going to only recruit carnivores versus vegans.
Stan Hazen did it at the Cleveland Clinic.
But I'm just kind of curious if there's
He looked at L-carnitine and red meat consumption. Is more like it wasn't a sepsis study, but just the L-carnitine pool and metabolism in vegans, vegetarians, and carnivores. And there's a difference, right? And
Why only 18? 15.
I don't know. I don't know the answer to that question. I was shocked when I saw 18 grams, Dushin, because that's a whopping dose. 18 grams of carnitine is a whopping dose. So just for context, L-carnitine really has very limited clinical use. It's used primarily to treat carnitine deficiency induced by chronic kidney disease. And so those doses, like if you look at those treatment regimens, they're pretty low. They use milligram levels. So,
One more question.
oh, one more question. Okay. Sorry for my living. Okay, here we go. Activated protein C has been found as there's death. Have you had a chance to test? That's a great question. No, because we haven't routinely measured or looked at activated protein C. It gets measured in some patients, but it got a bad rap after the failure of that clinical trial. And so I think that it just has kind of fallen out of favor, both as a biomarker and as a drug target. So we haven't like categorized patients based on their inflammatory status. So just to put this in context, I started my talk by telling you the massive number of patients who get sepsis and what a significant health hazard it is. The RACE trial enrolled 250 patients. 250 patients. Like, that's a small clinical trial. So we're really limited and we've got a very heterogeneous disease, right? So that's a really small clinical trial to start piecing out these different measurements. So we just need more patients. We need more patients that are have a lot of phenotyping, a lot of biospecimens that we can start looking at.
Alan has a question.
And Alan was and the metabolism pathway be channeled to the lysosome to break down sepsis. Alan, I don't think so. I mean, the lysosome is the garbage can, so there's a lot of stuff that goes in there, but I'm not sure how it might alter metabolism specifically.
I think that's all. Any question from the audience? I think that's all. Oh, yeah, Jose.
Great talk. A question related to the race study. So it's very interesting to see, acetylcarnitine can be used as biomarker to identify patients might benefit from treatment, right? So you come up with a cutoff value. I can't remember exactly. It's like 34,
30 micromolar.
30 micromolar. So it's curious, how you identify this cutoff. A follow-up question is, so besides identifying those might benefit from treatment, we'll also use different value to determine different doses for treatment.
Like different dose ranging based on their
Exactly.
levels. Yeah. So for the kind of acetylcarnitine cutoff value, if you will, we just did a series of threshold tests based on looking at mortality, placebo versus the 18-gram group. And we just watched the mortality improve as the acetylcarnitine level went up. And once we hit significance on that acetylcarnitine value, we said that, okay, like if patients had that value, they would potentially derive a benefit from supplementation. So that was done statistically using Z-score ranging for all the patients in the metabolites in the cohort.
Good, thank you. Let's thank Kathleen again.
Thank you.